Uh, Hello everyone. Actually, we are experiencing some delay with our speaker. So we kindly request you to be there for a couple of more minutes, and we hope that the technical issues will be sorted out soon. We'll get back to you. Yeah. Okay. I got response from her postdoc that she's joining in a couple of minutes.
Okay, okay. Great. Thank you, Saurabh. We'll hold on. Saurabh, you want to just make an announcement that she's joining?
Yeah, hello everyone. It was just we got confirmation that our speaker will join in a couple of minutes. We are really apologetic for this, but please hold on for a couple of more minutes for this. I'm sure you will enjoy the her talk a lot,
Sounds
and
good.
it
And
will
we
be.
promise to end on time. Okay. All right. So, Rohini, once the speaker joins, you can start the introduction. Hand over to Saurabh and Deepa. They'll introduce the speaker. She'll give her talk, and then question and answers. Sounds good? Yeah. Just make sure that the speaker can screen share and all, Rohini. Yeah, she's joining.
Yeah.
Hi, Elly.
Hi, Elly.
Hello,
I think
Elly.
you're muted, Elly.
You are muted, Elly.
Elly, you're muted.
Okay, apologies. I was at a meeting that went 40 minutes over and I didn't realize that it was going over. Okay.
Yeah.
No worries,
No.
Elly. Welcome.
Okay.
So without any further delay, probably, and I would like to introduce our today's speaker, Elly, and I've been knowing Elly for a few years now, and it's been amazing experience just talking to her. Just always like she's such a cheerful, and also like has been like a very prominent scientific figure in the field of regenerative biology, and it's been always great to hear their research journey. Let's not waste time, more time, and enjoy listening to Elly's research journey. Thanks, Elly. Yeah.
Okay. Thank you very much. So let me find my living. Okay. Is that correct? Is that
Yeah,
correct?
that's perfect. That's perfect.
Okay. So I'm really happy to give this lecture. If I understand, it's to a lot of young scientists, maybe even undergraduates. And so, I wanted to talk not necessarily about even the, most recent research, but, the research we've been doing over the years and kind of some of the interesting experimental and other aspects that have come up as we've tried to answer questions that we think are important to understand regeneration. So, um, um, right. So we work, of course, on regeneration in salamanders that regenerate the limb. Just a sec, for some reason, this just a sec here. Okay. So the model organism that we work on is Ambystoma mexicanum, which is this animal here. And it's able to regenerate many different body parts, including the tail and the jaw, the gills, and of course the limb. And pretty amazing that you can cut the limb either in the upper arm or the hand, and then it regenerates only the missing part. And it does it by recruiting cells from the stump tissue to generate a zone of stem cells called the blastema that undergo all the growth and patterning required to replace the missing part of the tissue. So today I wanted to talk about this issue that we've worked on for a long time, which is where do the cells for regeneration come from, and how do they turn into these regenerative stem cells? And before I start going into the specific experiments, I just thought that, to try and convey that it's been a very exciting time to work on such a model organism. Regeneration was a bit hard for people to apply molecular technologies when, the explosion of molecular biology happened in the kind of 80s, between the 60s and the 90s because you're working with adult tissue. So, there was a lot of work in molecular embryology in the 90s, 80s, and 90s, where people could make perturbations in eggs and then look at phenotypes in developing animals. But here you're starting with an adult tissue that's already very complex with many cell types there. And so how to, apply molecular techniques in an adult tissue, but also in an organism that not so many people worked on, was kind of a significant issue. But over the years, we were able to develop all the methods that people do use to study tissue biology on a molecular genetic level. And as I'll talk later, we could make germline transgenic animals, which has been an extremely important aspect of being able to study regeneration. And then, of course, with the recent amazing developments in genome editing, we can now knock genes into the locus to even tag proteins in the axolotl. This has transformed our ability to test gene function during regeneration. We can also mutate genes through injection of the CRISPR-Cas9 complexes into the egg to study phenotypes. But we can also express genes in the regenerating tissue in a localized way through the use of electroporation, coupled with live imaging, to understand, how genes affect the way cells function during regeneration. So this combination of gene perturbation in the adult tissue, coupled with the ability to follow the cells from time point to time point during the regeneration process, has really been very important for us to understand the process of regeneration. Then, in addition, as with the mammalian systems, we've been able to develop several viral systems, viruses that are able to infect mature and regenerating tissue. And here's an example where we expressed the sonic hedgehog gene in the side of a limb together with nerve deviation, and then we can induce an entire limb to form. And then finally, very exciting these days is through all of the developments in next generation sequencing, it was finally has been finally possible to assemble the axolotl genome through long read sequencing and the collaboration with very smart mathematicians, genome mathematicians that understood really how to take the inaccurate PacBio reads to assemble the axolotl genome, which is 10 times the size of the human genome and contained many repetitive sequences. So had been basically impossible to assemble in the early days of Illumina type-based sequencing. So we're very fortunate that over the last 20 years, the parallel, our own efforts in the system, as well as parallel developments in general for genome manipulation, have really opened up the possibility to study regeneration in an animal like the axolotl. So, I wanted to start with a very old observation that was sitting in the literature and that I wanted to tackle since my postdoctoral days. And this was the observation from Elizabeth Hay that showed that when she studied newt limb regeneration, so the newt is a different type of salamander that also regenerates its limb. She, at that time in the 1960s, the incorporation of tritiated thymidine was a new method that came up in the field. And so she applied this to regeneration to look at where's the location of cells that are undergoing DNA synthesis. And she came up with a surprising finding that in the transition zone between the stump and the regenerative blastema, she found what looked like muscle fibers that have incorporated tritiated thymidine in their nuclei. So, and then here you can see what looks like a fragment of a muscle cell that has incorporated tritiated thymidine in its cell. And so she made the kind of heretical proposition that during regeneration, differentiated muscle cells are dedifferentiating to form blastema cells. Now, so this is the proposition that you cut the limb. And actually, before I move on to that, I think the regeneration literature has been going on for 200 years. And there are many very interesting observations that people made with the techniques that they had at that time in, the early 1900s, the 1960s, that are worth revisiting with molecular techniques. So regeneration is one of those fields with a very deep history. And by knowing the old literature and looking at the old literature with young eyes, I think there are a lot of very interesting biologies to understand. And so I really encourage people to read the classical literature of fields like regeneration to get inspiration for what are deep and interesting questions and biologies that are worth tackling with modern methods. So to summarize then, the proposition is that you cut the amputated limb and these muscle fibers are dedifferentiating. They're generating mononucleate cells which proliferate and then regenerate muscle during regeneration. But in the meantime, then in birds, frogs, and in mammals, people identified a putative stem cell and now really a confirmed stem cell for muscle. It's called the satellite cell that sits underneath the basal lamina, coating muscle fibers, and these cells express the transcription factor PAX7. And upon muscle injury, these PAX7 positive cells proliferate, and then they fuse into the injured muscle in order to repair this muscle. And so in multiple species, it was clear that there's a stem cell that exists in order to rejuvenate muscle. So then what's going on during limb regeneration? In essence, are you taking a stem cell and then driving regeneration through the activation of a stem cell that differentiates, or could regeneration actually be the backward step, as in muscle dedifferentiation of a differentiated cell taking on progenitor cell capabilities, proliferating, and then redifferentiating in order to regenerate the structure? So Andras Seaman first was able to show by PAX7 staining in newt limbs that newt muscle does have PAX7 positive satellite cells. So there is a stem cell pool. And so it really people were wondering, okay, do you have both mechanisms, muscle dedifferentiation and stem cell activation? Or was there something, the conclusion from Elizabeth Hay was, did it have some false assumptions? And so interestingly, Andras in his lab and using the newt, and me in my lab using the axolotl started to try and look into these issues, whether there is muscle dedifferentiation during salamander limb regeneration. And muscle is a great system to ask whether there's dedifferentiation because there's such a clear transition of a multinucleated differentiated fiber breaking apart into mononucleate cells. So even if you don't know any, molecule that might be a marker, you could just look by morphology whether this dedifferentiation event was happening. So in order to do that, we felt that we had to reliably be able to label a muscle fiber and then be able to trace its descendants in the regenerating tissue. And by now, I guess many people have heard about what can often be called genetic fate mapping, and that's dependent on the use of Cre and LOXP. And so this is a typical genetic cassette that people use for genetic fate mapping. It's a ubiquitous promoter. Then you have a LOXP, a landing site for recombinase. EGFP, and then 3 polyadenylation sites that prevents transcription through this transcript. Then you have a LOXP and then cherry. And so these LOXP landing sites are arranged in a way so that when the Cre recombinase induces recombination between these 2 LOXP sites, the EGFP polyadenylation site is removed and the cherry comes next to the CAGS promoter. And since this is now constitutive, you get cherry expression in any cell that initiated this recombination. So, and then this is so we made a transgenic animal that has this construct in it. Sherry Arkatak was a really great postdoc who initiated this work. And then in this particular case, we're making a double transgenic with a constitutive promoter. And then this is a Cre that's inducible by tamoxifen, the hormone tamoxifen, so that in a certain time, we can cause this Cre to be active, and then it starts acting on this, and then we get cherry expression. So this is tail tissue that, this is a bright field image. This is the GFP image. And then you can see before tamoxifen, it's cherry negative. And then after injection of the animal with tamoxifen, then you see the expression of cherry. And you can see actually that there's still GFP in the tissue. You might be wondering why. And this is probably because these are muscle fibers and they're probably multinucleate. So that meant that, in some nuclei, you got this recombination, in other nuclei you didn't. And so you have co-expression of GFP and cherry. Or you might have multiple cassettes integrated into the genome. Now, this is a constitutive promoter driving the Cre, but what we wanted to do was to drive the Cre behind a muscle-specific promoter, so that in the mature limb or the tail, we would induce this recombination only in the muscle cells. The muscle cells would start expressing cherry, but they wouldn't turn it off if they dedifferentiated. So that then we would be able to trace the cherry cells throughout regeneration and then ask, do we find proliferative cells in the blastema? And then do they redifferentiate into muscle? Do they redifferentiate into something else? And so that was the big plan. And what was amazing is that Sherry made these transgenics and showed all this. But then whenever he tried to make a Cre construct driven by a muscle promoter, we could just never get a transgenic somehow. And we struggled for quite a while to get any kind of transgenic where muscle was driving this inducible Cre construct. And, we now have actually Prayag Murawala, postdoc in my lab, managed to get such a thing many years later. It turns out that the Cre-ER construct is very easily silenced in the genome. So we were probably getting transgenics, but they just were not being expressed. And so it took us quite a while to understand how to make transgenics with the inducible Cre-ER. But in the meantime, we were getting pretty desperate. We really wanted to answer the question of how does a muscle fiber dedifferentiate during regeneration. And, one day just sitting on the bus, I was talking to somebody about my problem. And then just, the light kind of turned on in my brain and I realized, okay, there's a way around this. And so the way around our technical problem was to actually use our CAGS Cre-ER construct, this one that expresses in all cells, and then also take advantage of the fact that muscle cells are multinucleate. They're the result of myoblasts fusing with each other so that you have cells, multiple nuclei within the same cytoplasm. And so based on that inspiration, what I realized is that we could also take advantage of the fact in the axolotl, you can graft tissues from one animal to another and they don't get rejected. So these 3 things we brought together, and we amputated the limb of this transgenic axolotl, CAGS LP cherry, and then we grafted the blastema from the Cre-ER, the constitutive Cre-ER construct, onto the stump. Oops. And sorry, this blastema should be over here. I don't know what happened here, but anyway, and then we allowed this grafted blastema to regenerate, predicting that this regenerate would be a chimera, a mixture of cells that came from the original stump and from the grafted blastema. And that since it would be a chimera, then the muscle fibers that formed during that regeneration might have formed as a fusion between myoblasts of this genotype and myoblasts of this genotype. So in essence, we're generating a double transgenic muscle fiber through fusion. And the prediction is that muscle is the only cell type in the limb that forms through cell fusion. And so then we would get specific labeling. So the idea would be to inject tamoxifen, and the prediction would be that we would see only muscle fibers turning red because they're the only cells that have both cells of both genotypes in the same cytoplasm. So this is what Tatiana Sandoval Guzman developed to do. So just to go through this again, the idea is that within the blastema, you have myoblasts of this phenotype, this genotype, and this genotype. They fuse together. So that means the Cre from this nucleus is going over there and then cutting out the GFP gene and then causing cherry to be expressed in this from this nucleus, and this cherry then distributes throughout the cytoplasm of the muscle fiber. And so here's one of these chimeric limbs that was made by this grafting of the two genotypes together. And then without tamoxifen, there's no cherry fluorescence. And then after injecting tamoxifen, we see nice, beautiful cherry positive muscle fibers. And so when we saw that result, we were just jumping up and down. It was just so amazing, so super happy. And just from these whole mount images, it looked like the cherry was only being expressed in the muscle. But of course, we wanted to check extremely carefully that the cherry was really only being expressed in muscle, because we wanted to see whether we got cherry positive blastema cells, and we wanted to conclude whether they came from muscle or not. And so there we used two molecular markers. MEF2C is a transcription factor that's only expressed in the nucleus and of muscle cells. And so you can see we have cherry. This is a cross section of one of these limbs, these chimeric limbs that had fully formed. And then we stained for MEF2C. And so then when we look here at the double labeling, that when you have a cherry muscle fiber, it's and you can see a MEF2C cell in there. It's the MEF2C positive nuclei is in cherry positive cells. So then we, on the converse side, we stained the tissue for the PAX7 stem cell marker. And since that cell should not be in the muscle fiber, it should not be cherry, and that indeed is the case. So you can see there's no overlap between the PAX7 staining and the cherry staining. So we convinced ourselves that the cherry expression was only in the muscle fibers. So then that gave us confidence to amputate those limbs. And then we amputated the limbs, and this is the result we got. And so this is the regenerated tissue after we amputated the limbs of such an animal. And to our great shock and surprise, we don't see fluorescence signal in the regenerated part of the limb. And this is a control where we hadn't transplanted the other genotype onto the host limb. And just by, we could quantify the fluorescence by taking a line scan of intensity here, and then you see the intensity going down. So although Elizabeth Hay had suggested that in the salamander, the newt salamander, there was muscle dedifferentiation going on, at least when we check in the axolotl by these transgenic means, we did not see evidence for muscle fibers dedifferentiating in axolotl limb regeneration. And, we were very careful. We took cross sections of the tissue. And so you can see here with this red staining that in the stump part of the tissue, we had plenty of red cells. But then in the lower parts of the regenerated parts of the limb, we saw no cherry signals. So quantification is a very important way to convince people that about your results. And so we conclude that in axolotl, we do not see evidence for muscle dedifferentiation. And in other work that I'm not going to show you today because I want to describe other things, we have experiments using transgenic animals that show us that PAX7 positive cells are the quantitative contributor to axolotl muscle regeneration. Okay, but what about this observation? And so this becomes the tale of two salamanders. As I told you, Anders Siemens' lab was attempting to address the same question in the newt. And while we can make transgenic axolotls because they have a generation time of about a year, it is long, but we are able to make transgenics. And once you get a transgenic, you get 500 eggs per laying. So we're really able to work with transgenics in the axolotl. Now, the newt has a different life cycle. It takes about 2 years to go to sexual maturity, and not only that, it has an aquatic stage, it has a terrestrial phase. It's very complicated to try and get newt transgenics. And so Anders Siemens' lab then used a different approach to bypass making transgenics to try and look at the same question. And there, as I mentioned electroporation before in the introduction, they electroporated the combination of 3 plasmids into the upper limb of the newt. And so that's, in fact, a very similar LOX-P type genetic fate mapping cassette here. And in this cassette, the cassette is flanked by Toll2 sites so that when they co-express a Toll2 transposase in the same cells, this will get integrated into the genome so that you have a stable cassette in the transfected cells there, and then a muscle-specific Cre protein, a Cre driving plasmid. And so then the idea is that you integrate this into the genome of the cells, and then this muscle-specific Cre is only active in muscle cells that get electroporated. So then you would get this expression of H2B YFP only in muscle cells. And so then when you amputate, you could follow these YFP nuclei in the regenerating tissue. And so this is the work from their lab. And in contrast to the axolotl, when they amputated the limb and then allowed it to regenerate and then looked at sections all along the limb here, they saw YFP nuclei in the regenerated part of the tissue. And so from their experiments, they concluded that muscle fibers are contributing blastema cells to the regenerated blastema that are then contributing to the regenerated limb. And they could also look, this is a longitudinal section of a regenerating limb. This is the muscle, and then these are the blastema, and you see these YFP positive cells in the blastema, and they could show that these cells incorporate EDU, a nucleotide reflecting S phase, so that these cells became proliferative. Or also they express PCNA. So here's the evidence that these cells incorporate BRDU, which is this also nucleotide analog, indicating that they had proliferated during this whole process. And then interestingly, in the blastema, when they look for PAX7 positive cells, they don't see any PAX7 positive cells in the blastema. So Andres's lab is arguing that the newt does undergo muscle dedifferentiation, and this seems to be a significant source of muscle in the blastema. So here we have 2 different conclusions in 2 different salamanders, but we wanted to be really sure to say that axolotl muscle does not undergo dedifferentiation and newt muscle does undergo dedifferentiation, because in the end we combined our results and published a single paper saying that there's differences in the presence of muscle dedifferentiation in different salamanders. And so we wanted to be really sure about this. And so then my lab went back, since we used two different techniques to label the muscle fibers. My lab went back, and actually I did a lot of these experiments myself, and went back and performed the electroporation experiments using the same plasmids in the axolotl to see whether we could, whether we confirmed our result of no muscle dedifferentiation in the axolotl. Because if we suddenly started to see, YFP nuclei in the regenerate in the axolotl, then it would mean that maybe there's something different about the way the methods worked. And so here you can see the result of such an experiment where we electroporated muscle with these three plasmids and axolotl, amputated, and looked at the regenerate. And indeed, we could see no evidence of YFP nuclei in the regenerated axolotl tissue. And so we quantified so that we could see 300, YFP nuclei in the stump tissue and then none in the lower arm and the hand. So from those results, we, conclude that there two different salamander species may use different mechanisms for blastema formation. In the axolotl, it seems to be that you form the blastema, PAX7 positive satellite cells migrate into the blastema and then contribute to the differentiating muscle. And in the newt, muscle fibers dedifferentiate, migrate into the blastema and contribute to differentiating muscle. At the moment, it's a little unclear what the PAX7 positive cells are doing in the newt, whether they just don't migrate into the blastema or whether they migrate and then they undergo forward differentiation and lose their PAX7 positive status and then start forming muscle. So that would require the genetic fate mapping of PAX7 positive cells in the newt. So that, I think that's a very interesting story of how there are multiple ways to regenerate and all the kind of technical aspects that we had to overcome in order to really study this process in the animal. So I don't know whether you want to spend the next period of time with questions. I can stop here, or I can talk until 12:30. And also, I'm happy to stay a bit longer. I'm sorry that I was a little bit late today, but maybe somebody can let me know what they prefer.
We can maybe do a short segment. We can continue your talk a little more and then take a few questions at the end.
Okay. So I wanted to talk about one other tissue that's very important in regeneration, and these are the connective tissue fibroblasts. And one reason why we think these are important is because of this issue. So, I told you in the very beginning of regeneration of the talk that regeneration is amazing, right? Because you amputate in the hand and then you regenerate the hand, you amputate in the upper arm and you regenerate the entire limb. And, from a conceptual point of view, how that happens was shown by the following experiment, where the hand was amputated and then the stump was grafted into the flank like this, making a circular arm. And then when Butler amputated such a limb, he generated a limb of the opposite polarity. But nonetheless, this limb regenerated lower arm and hand. So this experiment indicated that cells at the amputation plane are not reading the vector directionality of the limb tissue, but rather they know that they're upper limb tissue, and something about the wound causes these upper limb cells to then form lower arm and hand. And so this was called the rule of distal transformation, that, the proposition is that cells have memory of whether they're upper arm, lower arm, and hand. And then the amputation plane can only convert them into more distal structures. And we asked then, using our transgenics, whether all cells in the limb have this positional memory, or whether there's certain key cell types in the limb that have this positional memory. And so we did this in the following way. we took an upper arm stump, and then we took a transgenic, which only expresses GFP in muscle. And then we made a hand blastema on that transgenic animal, grafted it to the upper arm stump, and then we know that a normal limb grows from such a graft, and that that the regenerating tissue is a mixture of cells coming from the stump and coming from this grafted hand blastema. And when that regenerates, you get GFP expressing muscle not only in the hand, but also the lower arm and the upper arm. So that means that these hand muscle specified cells don't obey the rule of distal transformation. Now, in contrast, if we do the same experiment with a transgenic animal that only expresses GFP in what would be called connective tissue, these are the descendants of lateral plate mesoderm in the embryo that form the bone, cartilage, tendons, ligaments, and dermis. If you do the same experiment of grafting a hand blastema onto the upper arm stump, and that regenerates, then amazingly, you only get GFP expression in the hand. So that means these lateral plate mesoderm descendants that form the skeleton of the regenerate do obey the rule of distal transformation. And so what we believe now is that these connective tissue cells that form the skeleton, basically template out the patterning of the limb, and that other cells like muscle cells and also Schwann cells don't have this kind of positional memory and are followers in this whole process. So this is one reason why studying connective tissue cells in regeneration is so important. They seem to be the very intelligent cells of the limb tissue. So Josh Curry in the lab made a transgenic animal where he used a rainbow construct to multicolor label these connective tissue cells. And then he wanted to live label to understand two questions. How do these cells migrate to the amputation plane? And then does a cell migrate to the amputation plane and become multipotent? Because connective tissue, as I say, is a set of multiple tissues: bone, cartilage, ligament, tendon. And we didn't know which of these cell types was making the blastema, dermis also, and then whether cells were interchanging fates between these different cell types within this connective tissue lineage. And so in order to perform live imaging and get samples that were small enough, he amputated the digit tip and then looked at the process. And so this is the process of a multicolored tissue regenerating. And so then he could see that the cells close to the amputation plane migrated across the amputation plane early into the tip of the blastema. And then these are, for example, color-coded two descendants from the original cell that had migrated in. And one is a periskeletal cell, and then another cell is cartilage. And he could see evidence for this kind of transition of the daughters for contributing to two different, at least two different cell types in the regenerating limb. The other thing that he found is that many of the cells at the amputation plane do migrate across the amputation plane and contribute to the blastema. So, for example, it's not the selection of a very rare stem cell that, hyperproliferates to make up the blastema, but rather many cells at the amputation plane are migrating to the blastema to contribute to the blastema. So, to summarize these results, these connective tissue cells here are labeled in green. They're kind of in the dermis, they're between muscle fibers, they're wrapping around nerves, they're wrapping around bone, and actually they're also part of the bone, the chondrocytes. And then the green cells, the chondrocytes don't migrate across the amputation plane, but rather in the bone, you get a callus that forms a little bit of bone. But most of the new bone that's being regenerated comes from these dermal and interstitial fibroblast cells migrating into the blastema and then regenerating the bone and other tendons and ligaments. So we became interested in this cell type. And, in parallel to Josh's studies, we were really trying to understand, okay, how do we show whether a fibroblast dedifferentiates and makes a multipotent cell or not? And, do we have to do clonal tracing and all this kind of stuff? And at that time, Barbara Treutlein established a group in an institute 1 hour away. And she had pioneered single cell sequencing in the context of lung development at Stanford. And, Prayag and Dunya in my lab became interested in really trying to use the single cell transcriptomics to phenotype fibroblasts during regeneration. And Barbara and her scientific and life partner, Gray, were interested in regeneration. And so they got in touch with each other and then said, let's look at regeneration using single cell transcriptomics. And if I'm really honest with myself, I have to say that I was a little skeptical, because single cell transcriptomics was known to be quite shallow. And I was wondering, I was just worried whether we would really see any kind of meaningful genes. I thought we would only see the most abundant genes like actin or whatever, and then we might not be able to get much information out of the whole thing. But they were super enthusiastic and went forward. And Prayag had made a very important connective tissue M-cherry driving reporter, again, based on the CreLox-P cassette and then a PRX promoter driving the recombination. And so that allowed us to specifically fact sort out these fibroblastic connective tissue cells from the limb tissue, and then do a time course of regeneration. And importantly, this time course was occurring at a density that was less than a single cell cycle between the time points. So axolotls have a very long cell cycle. And so that meant that there was not a lot of proliferation between the time points. And in addition, not shown here, is we profiled developing limb bud cells. And so you can see here that these are single cells that as the mature cells go into the blastema, they get closer and closer in transcriptional space. So these fibroblasts are sitting in different niches in the mature tissue, and then they're all converging to a blastema-like state. So their transcriptional divergence is going down, and this is also reflected in this kind of depiction of the time course and the single cells. So, other analysis that they did allowed them to show that the fibroblasts, as they go into the blastema, take on the signature of a limb bud progenitor cell. And this will come up in next slides. And they cease expressing fibroblast ECM genes. So in essence, through the transcriptional profiling of these time points, they saw that fibroblast cells were then turning into cells that turn on MMPs, and then they started turning on blastema and proliferation genes. And then when they looked at limb bud cells, developing limb bud cells, they could identify the signatures of transcription factors that are involved in the development of the upper arm, the lower arm, and the hand regions, as well as the anterior and posterior part of the limb. And when they looked at the blastema cells at 11 days after amputation, they saw the re-expression of this set of transcription factors in the blastema cells after they had gone into the blastema and been there for 11 days. So the hallmarks of regenerative fibroblasts is that in axolotl, these cells do dedifferentiate to make a multipotent limb bud-like cell. And also that fibroblasts that come from the upper arm go into the blastema, and then obviously they're able to convert into a lower arm and hand cell. So they wanted to know, what about non-regenerative animals? What we wanted to know is, what about non-regenerative animals? So we decided to look at the frog here and to compare regeneration in the axolotl with the frog, which when you amputate the limb grows a spike of cartilage, but does not regenerate an entire limb. And so this is the work of Xiang and Yuka, and again, collaborating with Barbara Treutline's lab. And so just to very quickly go through this, this is the single cell profiling, again, of single cells across a time course of regeneration. And then here are the single cell profiles of limb bud cells. And so, so what Barbara and Toby were able to do was to identify a set of genes that's characteristic for limb bud cells, and then a set of genes that's characteristic for mature fibroblast cells. And then they could ask during regeneration, what fraction of these genes are expressed in the blastema cells? And you can see that the axolotl blastema cells become very close in their transcriptional signature to developing limb bud cells. Now, in contrast, when we do the same experiment in the frog, here's the limb bud signature, here's the mature fibroblast signature. They don't go up nearly as high as the axolotl cells. So that means that their dedifferentiation is somehow limited in the frog. You don't get complete dedifferentiation of frog fibroblasts. And then the question came to us, is that because of the environment of the adult frog, or is that some kind of intrinsic block inside the frog fibroblasts? So in order to test that, Xiang took made transgenic frogs and then took transgenic frog fibroblasts and also blastema cells and tried to test their potency through a heterochronic transplantation into the frog limb bud. He's basically giving these cells the most permissive environment to be a frog limb bud cell if they could and wanted to, and asked, okay, do these cells contribute to the skeleton? And from an upper arm blastema, would they be able to contribute to the foot? And in order to validate this assay, we transplanted frog limb bud cells to limb bud cells. We should be able to see cells in cartilage there. And then we also did the same experiment in axolotl. We took axolotl blastema cells and axolotl fibroblasts and transplanted into an axolotl limb bud. And that should also work. Otherwise, our assay is not valid. And so here's the limb bud to limb bud transplant in Xenopus. This is the limb that developed after that transplant. And these are the cells. This is a huge swath of cells that integrated into the tissue. And when he sections this foot area of the tissue, he sees transgenic cells that have incorporated into the cartilage and express SOX9. So that means we're able to transplant cells into the limb bud in a way that cells contribute to the cartilage. Okay, so what happens now when we take post-metamorphic adult cells from the frog and put them into the limb bud? They do graft into the tissue. They do go to the foot, but then when Xiang sectioned the tissue, he did not see cells contributing to cartilage, but rather these cells contributed to what looked like dermal structures. So you can see the results of the transplants here. In axolotl, when we transplant limb bud to limb bud, we get about 20% SOX9 positive cells. When we graft blastema to limb bud, we get about 20%. And when we graft fibroblasts to limb bud, we also see the generation of SOX9 positive cells. Now in the frog, when we graft limb bud to limb bud, we see SOX9 positive cells, but when we transplant blastema to limb bud, we see no SOX9 positive cells. And then as a control, when we graft blastema to blastema, then we see plenty of SOX9 positive cells in the blastema. So we wanted to look at this on a molecular level, and this is the last little bit of data. So what we did was to ask, okay, transiently during this whole process, maybe these cells started to dedifferentiate, but then something weird happened. And so we took a GFP-expressing transgenic animal, a Xenopus animal, took the blastema, and then grafted into a cherry-positive hind limb host. Then we dissociated and then performed single-cell sequencing to compare the transcriptional profile of the host limb bud and the transplanted donor blastema cells. And here you can see the UMAP from this kind of data. Here's the distribution of the host limb bud cells. And here's the distribution of the donor blastema cells. And then here you can see the transcriptional profile and the clustering of the transplanted blastema cells, the cells transplanted into the limb bud. And so you can see that they remain close to the phenotype of the donor blastema cells and don't take on the genetic profile, the transcriptional profile of the acceptor limb bud cells, confirming that somehow the frog post-metamorphic cells are somehow either blind to the signals or locked into their identity and don't seem to be able to de-differentiate into a limb bud-like cell. So now Andre in the lab is working on CRISPR-Cas9 screens of axolotl fibroblasts to try and identify really the key components of the pathway involved in de-differentiation of these axolotl fibroblasts. So he's going to transduce with the viruses, the Cas9 and the gRNAs, a gRNA library, and then he'll transplant the edited cells into the limb and then look at phenotypes in regeneration. And he will look for gRNA enrichment or de-enrichment, and then look at the molecular phenotype of the cells that are undergoing the genome editing. So I want to stop there and say thank you for inviting me to this lecture, and I'm happy to take any questions.
Thank you so much, Elly. That was a fantastic talk. We do have quite a few questions, and I will read some out. So some of the questions sort of deal with looking at PAX7, right? And, you know, is PAX7 temperature sensitive? You know, that's a question we've got from Satyanarayan, who wants to know if, you know, would this be crucial in higher altitudes?
Yeah, no, that's an interesting question. I mean, these animals especially are cold-blooded, and so they do change their phenotypes at different temperatures. We have not looked at that. You know, the animals do grow more slowly at lower temperatures, but whether the gene expression and, let's say, the rate of translation of factors like PAX7 goes down, we have not actually checked.
And is there any idea on what encodes the actual positional memory in the different cells along the arm?
Yes, so we have studied that. I didn't talk about it today. So what we've done is epigenetic profiling. So there's two answers to that. So we've recently done epigenetic profiling of upper arm, lower arm, and hand fibroblasts. And strikingly, what we see is that at homeodomain genes, those homeodomain protein genes that control limb development, they show differential marking with H3K27 trimethylation, which is a repressive histone mark. So in the upper arm, the hand homeodomain protein gene repressive mark, whereas in the hand, the MEIS homeodomain protein is coded with, which is an upper arm homeodomain protein is coded with this repressive histone mark. And so we think that these fibroblasts are poised in different chromatin states that then cause you to launch a hand development program in the hand and an upper arm development program in the upper arm. In addition to that, there are kind of cell surface molecules that Jeremy Brox and Max Yoon have identified that seem to be differentially expressed in the upper arm and hand and control the ability of cells to home either to the upper arm or the hand.
Great. We're just going to take two more questions in the interest of time. One of them is, do muscle cells and fiber nuclei in the axolotl express MSX1 during regeneration? Yes,
we think that most of the MSX1 is being expressed by the fibroblast descendants. I have to admit, I'm not sure whether the muscle descendants are expressing MSX1. There is this work from Mark Keating who in vitro showed that MSX1 seemed to be able to cause mammalian muscle fibers to de-differentitiate. I think it's unclear how many people can repeat that result, and we also don't know whether endogenously those cells are expressing MSX1.
And the last question is, have you also looked at expression levels of different developmental pattern formation genes? And did they also show changes during regeneration?
Yes. So actually, yeah, that was a whole nother lecture that I would have loved to give. As I mentioned, at the chromatin level, these homeodomain protein genes are differentially marked by this repressive histone mark. And when we look at HOX-A13 by antibody staining, if you make and HOX-A13 is associated with development of the hand. And actually, when we knock out HOX-A13 in axolotl, you get a very deformed or almost no hand. And when you amputate in the upper limb and form a blastema, you don't see HOX-A13 until like 11 days after regeneration, and only at the tip of the blastema, so the part that's going to form the hand eventually. In contrast, if you amputate in the hand and form a hand blastema, the HOX-A13 shows up much earlier at day at day 4 or 5, which is essentially immediately for an axolotl, and it seems to bypass the whole upper limb program. And so you have a different pattern of these homeodomain transcription factors depending on whether you amputate it in the hand or amputate it in the upper arm.
Okay, Elly, one last thing. Sorry for taking. The one message you would like to give to all undergrads and some, like, I know you shared it with me about exploring multiple possibilities and hopping around as much as possible, but yeah, if you would like to share something with undergrads and new researchers in India, it would be great.
Yeah. I think that now is an incredible time in biology. As I mentioned, there's so many ways you can manipulate biological systems through genome editing, through sequencing their genome. And, there are, so what's opening up now is the ability to study many aspects of biology and many organisms in biology that didn't exist before. And so I think exploring your ideas, as I said, reading the old literature of naturalists who are looking at nature and finding interesting aspects of nature can now really be re-explored using these biologies. And so, when I was studying cytoskeleton in my graduate school, and I was thinking about what I was going to do as a postdoc. And, I could have gone on to another lab studying cytoskeleton. But really, when in our graduate school, we were encouraged to think about working on black box problems, something that's really not understood. And, regeneration seemed to me one of those really fascinating questions at that time where very little was understood. And there were only really very few labs working on it. But, I took the chance to go to one of these labs who I, thought was doing super interesting work. And it was a bit of an unusual choice, but but because there was so little known, if we could make the progress to, find something new about it, I think it was really highly appreciated. And so I think taking the chance to study something you're very interested in where there's not that much known might be a little bit scary, but I think it's worthwhile. Yeah.
Great. Thanks, Elly. So thanks, Elly, for the amazing talk. And to all the panel attendees, like, we would like to end our season with this talk, like, great talk. We started with a great talk. We had fantastic talks in the series, and we will come back with the new dimensions with more speakers and with variety of fields in the next few months. But please check out for our advertisement over the social media. Thanks, Elly, again, and thanks all to India Bioscience for all their support, and Deepa for co-organizing this week.
Thank you
Thanks.
so
Okay.
Thanks a
Thank you
lot,
very
Elly.
much.
Thanks a lot, everyone.
Rohini, you just conclude.
Yeah, On behalf of the entire India Bioscience team, I would like to thank Deepa, Saurabh, and Elly. Thank you, Elly, for your insightful talk. Thank you, everyone.
Thank you.
Thanks a lot. Have a good day.
Bye-bye.
Bye.
Thank you. Bye.
Bye.
Bye.